everything goes ideal in the experiment optimal conditions, it can be concluded that worms are capable of associative learning by conducting various experiments to learn about the phylogeny of learning. Scientists have discovered that worms or planarians are capable of associative learning. Much later, learning has been demonstrated in octopus, mollusca, cephalopoda, which has a well developed eye and elaborated brain. Octopus can readily learn to discriminate the presence of white card signifying electric shock. First, the octopus was trained to come out of the hideout to seize a crab. After half the trials, a white card was lowered with the crab. This time, the octopus was driven away by the electric shock. After 12 trials, it started inhibiting its approach to the card, but after 24 trials, it constantly retained or remained in its nest when the white card was shown and come out to field when only crab was shown. This kind of discrimination has also been demonstrated in various other experiments, but problem solving has quite poor abilities. At the level of arthropods, much evidence of good learning is available. The ants, which are better learners than cockroaches, do not show generalized relearning when the maze was turned upside down and the ants had to run from the fruit to the nest. They had to learn the true path again. As compared to vertebrates, the higher invertebrates have relatively poor developed learning capacity. They are quite stimulus bound and their behavior is dominated by stereotyped patterns like taxes, reflexes, and instincts. All the different types of learning process deals in this chapter are supported mostly by the instances of vertebrates, more of mammals, less of fish and birds, and the least of reptiles and amphibia. Fish have been successfully trained in a variety of discriminations involving simultaneous choice between two different objects. They have mastered mazes and solved problems much more rapidly than invertebrates, except ants. Modifiability and ability to profit from experience are clear characteristics of these animals, but by and large, their learning is chiefly of trial and error type. Birds possess some rapidly and consistent learning. In mammals, further improvements in learning are apparent when we compare the rats with primates. At this level, the animals are increasingly free from simple stimulus response or trial and error learning. The past learning experiences are utilized in new learning tests. They bring the solution of new and complex problems by reasoning. Man, of course, is with highly evolved capacities utilizing past experiences readily and extensively. Though a comparison of learning abilities in different animals is still difficult, yet it is clear that learning ability is improving in phylogeny. We can roughly equate this increase with the evolution of large, not in terms of size but complexity of brain. Again, within the vertebrate series, the most dramatic aspect of brain evolution is the growth of the cerebral hemispheres, especially the cortex region, which reaches the climax in primates. Complex modes of behavior become possible in phylogeny. They are first added to the simpler modes but eventually replace them. At each new level of complexity, new behavioral properties emerge. Primates have new modes of adaptation which overshadow the simpler ones. Now we are gonna learn about the neural mechanisms in learning. From the above discussion of phylogenetic consideration of learning, it has become obvious that it is the evolution of nerve system which integrates the learning process. It can also be safely said that learning presents a change in the central nerve system and that memory is the preservation of that change. So, what is learning? Learning represents a change in the central nervous system, and that memory is the preservation of that change. We are preserving that change in the memory form. Two important questions are thus investigated variously just to tell the involvement of central nervous system in learning. Two important questions are thus investigated variously just to tell the involvement of central nervous system in learning. Which are those two questions? Where does learning take place in the nervous system? What is the locus of learning? What is the nature of change? So now let's learn about the locus of learning. The first question has been approached by experimentally destroying parts of nervous system, chiefly the cerebral cortex. Pavlov was of the opinion that the cortex is essential for conditioning, but on the other hand, studies have also shown that simple conditioning is possible in the dog even after its cortex was removed. So we are going to learn about the mammalian cerebral cortex in detail. Since most of the findings on neural mechanisms of learning are based on mammalian brain, it is essentially to have the knowledge of the later from anatomical point of view, but also because cerebral hemispheres are thought to be the site of memory and learning. These are mainly to be understood. Here, details of only cerebral hemispheres are taken up for further understanding of learning. The cerebral hemisphere develops as a pair of hollow vesicles from the roof of the forebrain, and in higher mammals, they are so large that they come to cover the entire brain. The walls of the cerebral hemisphere are called the cerebral cortex, which are thin, and the area of cerebral hemisphere is greatly increased by convolutions on their surface. Convolutions on their surface. The development of cerebral cortex is presented. The oldest parts of the cortex, paleopallium and the archipallium, are largely olfactory and visceral in function. A new cortex of neocortex appears for the first time in reptiles as a receiving area for sensory fibers from lower regions of the brain. In man and other specialized mammals, neocortex makes up the greatest proportion of the brain. A great tract of the nerve fibers, the corpus callosum, connects the neocortex of one side of the neocortex of another side. They are connected by the corpus callosum. Nerve fibers connects the neocortex of one side with the neocortex of other side. With the expansion of neocortex, two things happen. The paleocortex or the paleopallium moves to a more ventral position, and the archipallium, now called the hippocampus, folds into the cerebral mass underneath the neocortex. That is the first case when neocortex expands. Now the second case, the neocortex expands so that it begins to fold in on itself, developing the fissures or sulci and the bulges or gyri that characterize the convoluted brain. Now, on the basis of major fissures, the cortex of man can be divided into major lobes. In the early mammals, the neocortex is primarily a sensory receiving area. In rabbit and rat, those cortex is convoluted very little. There are fairly well organized and somewhat segregated areas for vision, hearing, somatic sensation, and taste that occupy most of the cortical surfaces. The motor area is situated anteriorly in the cortex. There are few and small areas in cortex which are neither sensory nor motor, called association areas. Very large in primates receive horizontal fibers from other parts of cerebral cortex, coordinating diverse sensory information. This clearly shows relatively small stripes of cortical fissure in man devoted to sensory and motor function, and most of the cerebral cortex is given over to large association areas. Despite their relatively small size, the sensory and motor areas of human cortex become highly organized and represent the functional parts of the body and the receptor surfaces. Now we are going to learn about the decortication experiments, which are conducted by restructuring or removing certain parts of cerebral cortex. These are called decortication. Decortication experiments. Many of the critical experiments on decortication effects were done by Carl Lashley. He started by training rats to run a maze. The trained animals were then operated, incisions made all over the cortex, cutting any chain of neurons running from point to point. These animals did not lose the ability to run the maze, and they were also able to learn the new mazes like normal rats. In this case, circuits across association areas could not be shown as important. Lashley then removed the motor cortex from monkeys trained to open latch boxes. The animals were paralyzed for 12 weeks. After the operation, when they recovered the ability to move, tests were again done, and it was found that they could open the latch box just as well as the unoperated control animals. This is suggestive that of the probability that the motor area is not needed for learning. In the next series of experiments, Lashley studied the effect of the total removal of slabs of the cortex on the ability of rats to learn to run mazes. These experiments showed that the defects in learning ability depend only on how much of the cortex is removed. Lashley concluded that the cortex operates on a mass action principle in learning and memory, so that the larger the lesion, the poorer the ability. The larger the lesion, the poorer the ability. Lashley also found that it did not matter that where the lesions was in the cortex, a lesion of given size had a given effect, whether it was in visual area or in the back of the brain or the somatic sensory and motor areas in front of the brain. From this, Lashley formulated another picture of equipotentiality, which states that all parts of the cortex are equal in their contribution to learning and memory. That is the principle of equipotentiality. In the animals, where the cerebral cortex is primarily sensory, it is believed that the larger the cortical lesion, the more it will impair the use of sensory cues, resulting in poor maze performance. Rats learn to use different sensory cues throughout the maze, like vision, sound, touch, smell, etc. The more the sensory cues are experimentally eliminated from the animal's use, either by destroying sensory organs or by removing stimuli, the worse its performance. But against this interpretation, Lastly taught blind rats maze running and then removed the visual cortex from exhibition of defects in the retention of maze habit. Concluded that visual cortex has non-visual functions in learning and memory, as well as visual phenotions. It also leads us to some reasonable conclusion that the cerebral cortex is not essential for learning and memory. All the cortical neurons work in groups or masses. Now let's learn about the subcortical structures and learning. Electrical stimulation of the temporal cortex of fully awake epileptic patients evokes past memories in a dream-like sequence, possibly as much through arousing subcortical structures as to the effect on the cortex itself. In one experiment, it was found that placement of irritative substance, aluminum cream, just below the temporal cortex in hippocampus was most effective in impairing learning. In other experiments, it was found that surgical lesions in the region of hippocampus produced the most marked defects in monkeys, which were required to discriminate two patterns of tone or of light separated by several seconds. The operated monkeys could tell that the two patterns were the same because the lesion made them unable to remember the first pattern over the time. Now let's learn about the electrical recording methods and learning. For a better understanding of brain mechanisms underlying learning, there has been another approach through the use of electrical recording methods in which it is possible to trace things in the electrical activity of many parts of the brain during learning. In this, the electrodes are chronically implanted in the brain of an animal, and the changes in pattern of electrical activity are noted as the animal is trained. It has been seen that changes take place in many parts of the brain, cortically, subcortically, within the sensory system, or outside them. This method has also led to the conclusion that learning occurs in many places of the brain at once, and that no part is completely essential for its formation or retention. So let's learn about the split brain technique. Function of cerebral hemispheres in memory has been investigated in greater details by Sperry and his co-workers. If the optic chiasma is split, things learned with the left eye are still known to the right eye, so long as the corpus callosum, the larger band of fibers which link cerebral hemispheres ventrally, is intact. As long as the corpus callosum is intact, when we separate the optic chiasma, things learned things learned with the left eye are still known to the right eye. If the corpus callosum is also separated, then one half of the brain no longer knows anything learned by the other half. The split brain experiments show that one function of corpus callosum is to transfer engrams or memory from one hemisphere to the other, and storage is eventually bilateral. Information flows in both the directions. This technique also reveals the role of isolated pieces of cortex in memory. If one half brain is left intact, it can serve to keep the animal alive and functioning well. Unless the brain is split, lesions made on one side only are useless because the other side takes over the whole function. Using this technique, it has been possible to show that different parts of the cortex are not equipotential as stated by Lashley. An isolated island of frontal cortex, together with the parts of the sensory and motor areas, can store the information or engrams for touch discrimination in the cat. The most important lesson to be drawn from the split brain animal experiment is that an operation can produce subtle yet reproducible changes in behavior. Dorsal view of a split brain shows the several optic chiasma and corpus callosum. When only optic chiasma is cut, things learned from the left eye are still known using the right, but when corpus callosum is also cut, no visual information can pass from one side of the brain to the other. Here we are talking about the visual information. Now, nature of change in learning, that is the biochemistry of learning. Various views have been expressed as being responsible for the establishment of new functional connections in the nervous system, the growth of new pathways in the nerves, anatomical swelling or sprouting of synaptic terminals resulting in the facilitation of cross synaptic connections, and biochemical changes like alterations in the structural arrangement of protein molecules in nerve fibers. Of all the suggestions, biochemistry of learning is now attracting a good deal of attention. The main reason behind this is because of the fact that in the nervous system, chemical changes are in some way easier to detect than structural ones. Like all the living cells, neurons are also continuously breaking down and building new proteins. As we all know from the physiology of neurons, transmission across the synapse involves synthesis of acetylcholine, a transmitter substance which diffuses rapidly through the synaptic membrane. Both synthesis of acetylcholine and its rapid breakdown after it has diffused through synaptic membrane require enzymes or proteins, and this extensive protein synthesis. Both synthesis of acetylcholine and its rapid breakdown require enzymes. Evidence suggests that during consolidation of memory or learning, the rate of protein synthesis is higher than for the normal needs of transmission. Procedures adopted to investigate role of protein synthesis in memory shortage are very similar to those used for physical disruption of consolidation. That means the procedures adopted to investigate the role of protein synthesis in memory storage are very similar to those used for physical disruption of consolidation. As an example, a drug, puromycin, that is a protein synthesis inhibitor, has been found to interfere with memory at varying periods after learning to run a Y-shaped maze, avoiding electrical shock and running to the left arm. The mice was injected with puromycin directly into the brain, controlled with saline. Results showed that puromycin injected mice show little sign of memory as compared to the control ones. If neocortex is involved while injecting puromycin, it is reported that the memory is lost. So that is something which is very shocking. Puromycin. Possible reason put forth by the investigators is that all peptide synthesis is hampered. Perhaps when training takes place, an essential RNA is manufactured which directs synthesis of protein necessary for preserving memory. In recent years, a good deal of speculations and experiments on the role of RNA in memory storage are available. Some of them can be summarized as under: RNA controls protein synthesis occurring during memory consolidation. The cell bodies of neurons contain large amount of RNA, more than that appear necessary for routine maintenance and impulse transmission. This does not explain those axons which contain little or no RNA. The cell bodies of neurons contain large amount of RNA, more than that appears necessary for routine maintenance and impulse transmission. The RNA in certain neurons are just trained. The RNA in certain neurons of just trained animals, such as rats, changes in its chemical structure as well as increasing in quantity. The quantity also increases when the animal or neurons are passively stimulated without learning. Injecting RNA extracted from the brains of trained animals into the brains of untrained animals which lack experience have been claimed to have reproduced the effects of training in the later. Conclusion: from the vast treatment of the subject of learning, it is evident that learning ability of animals is related to the development of the central nervous system in phylogeny, but it has not been easy with great specificity to discover the neural basis of learning. The evidences from experimental brain lesions and from studies that record changes in electrical activity of the brain during learning is suggestive of the fact that learning takes place in many places within the brain at once, although biochemical approach to learning has appeared convincing. But now, neurons come to change their functional interconnections as a result of experience is yet to be explained. Biochemistry may provide only subsidiary answers, and thus much more neuroanatomy and physiology need to be thoroughly investigated. In the context of behavioral principles, it is clear that learning is a peculiar effect of stimulative experience. Learning process itself may be an experience that can modify future learning, and is animals have to learn to learn.